So the project we did was nitrite poisoning, which in the past was the blue baby syndrome. But for this presentation, or this project, it's dealing with fish. So what happened was a man came home and he noticed that the fish in his fish tank were displaying different symptoms. One was rapid gill movement. The fish were hanging out by the filter in the water, and the gill color had like changed to being brown instead of whatever the color of the fish were. And he was trying to figure out why. And a friend of him mentioned brown blood disease in fish to him. So he decided to test it to see if this is what caused it. And what brown blood disease in fish does is it can be caused by overfeeding, or if you're cleaning out the tank too much, which causes a lack of bacteria in the water. And the bacteria takes ammonia, which is excreted from the fish through their waste or food, and changes it into nitrate, and then into nitrite. And if there's like not enough bacteria and they get overwhelmed, then those levels build up and become toxic. Or if there's no bacteria, then it becomes toxic. So the experiment he decided to do was to take 2 fish tanks with completely similar scenarios, except for one had bacteria in it and one did not. And so his hypotheses was that if the tank containing bacteria came up, or if the tank with no bacteria came up high, that means it was the lack of bacteria that caused the toxic levels. But if the tank with the bacteria came up high, that means there's too many fish causing the bacteria to be overwhelmed. So,
and the materials that were used for the experiment were the 2 fish tanks, which were of equal size, and one without the bacteria, one with. And then we had, and that was called the nitrosammosis. I pronounced that wrong. And then the other part of the material that we used was the tap water, the test tubes, the standard samples known as the nitrite concentration, and we also used the disposable pipettes and the spectrophotometer. And then we also used a color indicator to help us indicate the changes. And as far as the fish tanks, we made sure that they were the same brand as well as the same size. And we also used the micro pipette, which was the 20 color of it. And then we used the crevettes and also gloves to protect our hands from all the different materials that we were using. Also, so we wouldn't infect any of our materials that we were working with. And so, for the methods, we used the sorry, I always pronounce that wrong. And we measured the OD absorbance levels of each of the samples. And we also measured out 1 milliliters of each of the known nitrate concentration solutions. And we used the concentrations going from 1, 2, 3, 6, 10, 20, and 40. And we labeled them into each separate tubes. And then for the spectrophotometer, we set that to the 550 OD, and then we used a blank curvette, a consistent one, and we made sure we had pure deionized water. And after we got done using it, we put it back into the spectrophotometer. And once we ran that, we made sure that we calibrated the machine. And then for the first sample that we had started off with, we started off at a concentration of 0, and we made sure that we carefully transferred it to the curvette. And then once that was done, that was to completion, we made sure that we wiped it down. And then we inserted the curvette back into the spectrophotometer, and then the absorbance, it was tested once again.
So these are our results, and this was tank A is with the nitrobacter, so the bacteria that's going to help keep the levels down. You can see A is below 1, and when you register 1 on the spectrophotometer, it becomes inaccurate over 1, and you can see B is relatively high. So A registered a correct absorbance, and the nitrite concentration came out about 4 parts per million, which is relatively safe. But for B, we had to do a dilution because it is above 1 too. So to get an accurate reading, we did a dilution, a 110 dilution, with 1 milliliter from tank B, a sample, and diluted it with 9 milliliters of water. And then we registered that again on a separate graph. And plot a line of best fit again. Again, this is going up only until 0.6 absorbance, including only those samples. And B is shown below right here at a nitrite concentration of around 2, but that's with the dilution. So if you take it to the counterfactor and multiply it times 10, it actually comes out to about 16 parts per million, which is unsafe. So it can be concluded that the tank with no nitrobacter, the bacteria, is going to be less beneficial for the fish.
For the discussion and conclusion, this is basically the nitrogen cycle. First, the fish eats the plants, and the fish waste are decomposed into organic substances, and then the bacteria nitrosomas converts it into nitrite, which is toxic for the species of fish, and so then the nitrosomas has then converted to ammonia, has to convert it back to nitrite, and then it can then release the nitrogen into the atmosphere, so that the cycle goes back again. And nitrite under 10 ppm is safe. So in the experiment, 2 fish tanks were set up to test for the levels of concentration, and sample A had no bacteria present, and sample B had the bacteria present. After comparing the absorbance of the 2 fish tanks, one with the nitrosomas bacteria present, one showed a higher concentration than the other, because sample A had only 4 ppm and sample B had 18 ppm. And since 18 ppm surpasses the safe level of nitrite 10 ppm, it was obvious that the sample B fish tank was the one that caused the high levels of nitrite concentration and brown blood disease. These supports the hypothesis because the lack of bacteria caused a surplus of the nitrite in the water, not the number of fish overwhelming the existing bacteria. So that explains why the fish were hanging out by the tank's filter, because they were trying to stay near the cleanest source of water. So the hypothesis stated earlier was that if the tank contained no bacteria resulting in toxic nitrite levels, then the cause of the brown blood disease is from low bacteria levels. So to fix the problem and to cure the fish of the brown blood disease, bacteria was needed to be added to the fish tank in order to remove the toxic nitrite, and that's what it looks like with brown blood disease, and that's what fish looks like with normal disease. So the learning outcome of this whole experiment was that dirty tank water is unhealthy for the fish's living environment, but when you over-clean a tank, and it can actually reduce the number of nitrosomas bacteria, which makes it unable to remove toxic nitrite and can cause